Hello, my dear students, and welcome back. I know, the board exams are right around the corner, and the tension is probably building up, right? But please don't worry. Your favorite tutor is here, and together we are going to absolutely ace this chapter. So, I've been reading all your comments, and so many of you are saying Chapter 12, Tools and Techniques, feels a bit difficult. So many new words, so many new concepts, but trust me on this one. It is one of the most interesting chapters once you get the hang of it. So let's try something different. Instead of thinking of this as a list of boring techniques, let's imagine we have a magical biotech toolbox, and each tool inside does something really, really cool. So in this quick explainer, we're going to open this box together. Ready? Let's do this. All right, first tool out of the box. Let's call this one our super eyes. I mean, when scientists need to look at something ridiculously tiny, like a single cell, what tool do they reach for? A microscope, of course. Now, this whole science of using microscopes is called microscopy, and it's all about two main words, magnification and resolution. Magnification is simple, right? It's just like zooming in on your phone camera. It makes things look bigger. But resolution, that's the real hero. It's what makes the image sharp and clear, not all blurry and pixelated. And you know what? There isn't just one type of microscope. It's like having different camera lenses. The one in your school lab, that's a bright field microscope. But then you have dark field, which makes things glow. Phase contrast to watch living cells moving around. And the king of them all, the electron microscope, which is so powerful it can see a tiny virus. How cool is that? Okay, so we've seen the cells, that's awesome. But now, what if we want to sort out all the tiny bits and pieces inside the cell? For that, we need to pull out the next set of tools from our box, the Great Separators. The first one has a big, fancy name, Centrifugation. But don't let the word scare you. You've seen this in your kitchen. When your mom makes ghee, she puts cream in the mixer and spins it super fast, right? And the heavy butter separates out. Centrifugation is exactly the same idea. We just spin things really, really fast to separate them based on how heavy they are. And this is the machine that does it. A centrifuge. You see, the samples go into those little tubes, which are placed in the spinning part called a rotor. You close the lid, set the speed. We're talking thousands of spins every minute and press start. When it's done, all the heavy stuff is packed at the bottom. Simple. Okay, our next separator is called Electrophoresis. Another big word, I know, but here's the simple idea. It's a running race. But it's a race for molecules like DNA. And the racetrack? It's a special jelly-like slab called a gel. Now, every race needs a good racetrack, right? Here we have two main types. For the big, chunky molecules like DNA, we use an agarose gel. But for smaller, nimbler things like proteins, we use a polyacrylamide gel or PAGE. Just think of it like this. Agarose has big gaps for the large DNA to run through, while PAGE has a much finer mesh for the tiny proteins. All right, exam time, pop quiz. This question comes up all the time, beta, so listen carefully. In this DNA race, to make them run, we apply an electric current. So why does the DNA always run towards the positive side? Think about it. The answer is because DNA itself is negatively charged. It's as simple as that. Remember that sugar-phosphate backbone and its structure? Well, those phosphates are loaded with negative charge. And what do we know about charges from physics? Opposites attract. So the negative DNA gets pulled towards the positive electrode. Easy peasy. And here's the photo finish. Look at that. So who won the race? The little guys did. The smallest, lightest DNA fragments zip through the gel and travel the farthest. The big, bulky fragments, they get left behind near the start line. And to see all this, we stain the gel with a special dye that makes the DNA glow under UV light. All right, let's reach back into our toolbox. The next tool we're pulling out, this one's a detective. A super smart one, in fact. Its name is ELISA, and its job is to find one specific molecule hiding in a huge crowd. I know, the full name is a total mouthful, enzyme-linked immunosorbent assay, but just forget the name for a second. The idea is brilliant. It uses special COPs, called antibodies, to catch a specific bad guy called an antigen. If the COP finds the bad guy, an enzyme creates a color change, that's it. This is the basic idea behind so many medical tests, like the ones for HIV. So how does this detective mission work? Let's look at indirect ELISA. Step 1, we stick the bad guy or the antigen onto a plate.
Step 2, we send in our first detective, the primary antibody. It will only stick if it finds its specific target. Step 3, we send in a second detective. This one is trained to find the first detective, and it's carrying a little flashlight, an enzyme. Step 4, we add a chemical that turns the flashlight on. If we see a color, mission successful.
Now listen to me very, very carefully, my dear bachcho. Look at this diagram. This process, this picture, it is a 3-mark pakka question. I'm telling you now. It comes in the exams very, very often. You have to practice drawing this and labeling it correctly. Promise me you will.
And now for the final and maybe the most mind-blowing tool in our entire toolbox. We've seen things, we've separated them, but what if we could actually read the book of life itself? What if we could read the DNA code? Well, that is exactly what DNA sequencing does. It's the technology that lets us figure out the precise, exact order of the bases, the A, T, G, and C, in a strand of DNA. We are literally reading the instruction manual of an organism, letter by letter.
So how on earth do they do this? It's a very clever trick, actually. They use special imposter nucleotides called ddNTPs. Look closely here. See how they're missing that little oxygen atom? Well, that's the spot where the next block is supposed to attach. Without it, bam, the process just stops. It's a chain terminator, just like putting a full stop at the end of a sentence. And in today's world, it's even cooler. Each of those full stop bases, A, T, G, and C, is tagged with a different color. A laser in a machine reads the color of the last base on millions of fragments. And look at this output. The computer just reads the colored peaks and spells out the sequence for us. Technology is just amazing, isn't it? Phew! Okay, that was a whirlwind tour of our biotech toolbox, but you all did great keeping up. So, just before we wrap up, I've made a little something for you. Your very own exam time cheat sheet to make revision super quick.
Let's just run through it one last time. What does each tool do? Microscopy is for seeing, centrifugation is for separating by spinning, electrophoresis is that race that separates by size, ELISA is our detective, and DNA sequencing is for reading the code. That's it. When you boil it down to the main job, it's not so tough at all, is it?
And with that, we've covered the most important tools from this chapter. Now listen to me. My dear Bachchan, you have prepared well and you know this material now. I want you to walk into that exam hall with your head held high and with confidence. You've worked hard and you are going to do wonderfully. All the very, very best from me.